So here I'm going to walk you through a really nice example of protein folding using hemoglobin protein. So this is put together by the labx platform. I'll provide the link in case you want to go back and look at it in more detail on your own. So here is the hemoglobin protein. It's an essential protein for transport of oxygen from the lungs to cells all throughout your body. So it's this protein that carries oxygen throughout your body. And it has this structure here that is required for the binding and the release of the oxygen gas molecules. So we're going to zoom in on this protein structure and talk about how that contributes to its function. All right, so there are four different polypeptide chains in hemoglobin. Two of them are these alpha globin polypeptides, here in kind of a brownish color, and then two of them in blue are going to be the beta globin polypeptide chains. So there are two different sequences of amino acids that go to make up this protein structure. So in order for the protein to function properly, or any protein to function properly, it has to be folded correctly. Sometimes this can happen on its own, just in the aqueous solution, and sometimes it involves the help of another protein called a chaperone protein. So what happens with a chaperone protein is a new polypeptide gets packed into a compartment within the chaperone complex. It gets enclosed in that space, and that provides it the appropriate environment to fold correctly. Once it's correctly folded, it can leave. Proteins that are folded incorrectly or misfolded can be redirected back to these chaperone proteins to help them fold better. So as we've already talked about, amino acids have a conserved structure that consists of a central carbon here on the amino acid attached to an amino group and a carboxyl group. There's also a side chain identified by this R group here that is different for each of the amino acids. So now these amino acids get connected to each other through a dehydration synthesis reaction to make a peptide bond. So there are 20 different amino acids, and they can be grouped based on their properties. As we've already talked about, you can have nonpolar amino acids that are hydrophobic. We can have polar amino acids that are hydrophilic and mix well with water. And then we can have amino acids that either have a positive charge or a negative charge on their side chain. Negative charged ones are considered acidic because they can lose a hydrogen ion. The positive charged ones are considered basic because they can accept an additional hydrogen ion. So protein folding at each level is represented by different models depending on how much information needs to be conveyed. So primary structure, where you're looking just at the order of amino acids, usually it shows all the individual atoms by letters, and all of the lines represent covalent bonds. These primary structures are identified by having an N terminus where the protein starts and a C terminus or a carboxyl terminus where the protein ends. As these sequences get longer and longer, sometimes rather than writing out that entire sequence with all of the letters, you'll use a wireframe model like this to show each of those amino acids within the polypeptide. Or if that is still too messy and too complicated, you might show it as this ribbon diagram to show how that protein folds. This twirling structure here is known as an alpha helix. So this leads us to secondary structure. Alpha helices and beta sheets make up secondary structure. And within these alpha helices and beta sheets, the primary structure amino acids extend from the N to C terminus. The shape of these, or how the alpha helices and beta sheets fold, is dependent entirely on the sort of three-dimensional shape of the amino acids that participate in it. These alpha helices and beta sheets are held together by hydrogen bonds between the water molecules. And we can see that here. So within this beta sheet, you can see the little dotted lines connecting the upper beta sheet to the lower one, where there are hydrogen bonds holding the two sheets together. The same thing is going to be true in the alpha helices. So these hydrogen bonds occur between the amide group of one amino acid and the carbonyl group of the other amino acid. Right. Additionally, there are some unstructured regions of the polypeptides as well, and these tend to occur between the alpha helices and the beta sheets, where it switches between them. So if we look closer at how these alpha helices are structured, in this alpha helix, the hydrogen bonds are forming between the carbonyl group of one amino acid and the amide group of the other one. And that other amino acid is about 4 amino acids down the sequence from that first amino acid. That holds it in this really nice tight spiral. Right. Additionally, on these, all of the side chains can form other intermolecular interactions because all the side chains from the alpha helices are pointing out from the helix. Right. Beta sheets also form these same hydrogen bonds between the carbonyl groups of one and the amide groups of the other. And in this case, the side chains, or the R groups, are pointing along the same line as the beta sheets. Beta sheets are therefore better for amino acids with smaller side chains. Ones with larger side chains, these R groups tend to get in the way and cause conflicts with the neighboring R groups across the other side. So here's a wireframe model of some beta sheets. And then sometimes those wireframe models get a little bit too complicated and we have to back out one more level of visualization to look at the tertiary structure. So tertiary structure is even more complicated than secondary structure because you have those secondary structures interacting with each other. In this case, all of the interactions in tertiary structure take place between the R groups or the side chains of each of those amino acids. So the tertiary structure of this beta globin polypeptide, you can see is primarily composed of a bunch of alpha helices all interacting with each other. So protein folding in tertiary structure is controlled by the chemistry of the interactions of the side chains. The goal of the folding is to reach the most energetically favorable or energetically stable conformation of the protein. So the shape that makes it the most stable. So a lot of different factors can influence the stability of the protein. These include whether the polar or nonpolar regions of the protein are in contact with water, how much space is allowed between larger and smaller side chains, as well as charge-charge interactions. So here you can see the nonpolar components of the polypeptide are folded into the middle of the polypeptide, whereas the nonpolar ones are in the middle of the polypeptide, all hidden from the water, while the polar amino acids are all around the outside interacting with these water molecules. So there are 4 types of chemical interactions that can occur between the side chains to help build this tertiary structure. All right, the first, so here this blue line illustrates the polypeptide backbone, or the backbone of the protein structure. The first component that's going to help the protein fold are going to be hydrophobic interactions. And you can see those hydrophobic interactions here in the box where you have nonpolar amino acids interacting with each other. These nonpolar amino acids want to hide from the water, so they're going to fold generally to the inside of the polypeptide. So here the water is not going to be able to reach them. The second type of interaction that can occur are hydrogen bonds or polar interactions. These hydrogen bonds are going to form between 2 polar amino acids and help to stabilize the 3-dimensional structure of that protein. The third type of interactions are ionic bonds. Ionic bonds are also going to be polar, and they're going to be a little bit stronger because they're formed between things with a fully positive and a fully negative charge to help stabilize that three-dimensional protein structure. And the fourth component, the strongest of all, are going to be disulfide bridges or disulfide bonds that form between two sulfur groups of two cysteine amino acids. So here you have two cysteine amino acids. Those two sulfurs are going to bond and make a disulfide bond, holding these two backbones of the polypeptide together with a covalent bond. This is going to be the strongest interaction of all. Right, so now as the molecule gets bigger and bigger and more complicated, we might move from looking at it as a wireframe model to a ribbon model, and then to a space-filling model, where we can see how much space it takes up in three-dimensional space. Right. The final structure of the protein, called quaternary structure, is the most complex structure of all, and it depends upon the tertiary structure. So for these, you've got multiple polypeptide chains all coming and interacting together. So not all proteins have quaternary structure, but any protein that has at least two different polypeptides, or two of the same polypeptides even, can form quaternary structure. So here for hemoglobin, you have an alpha globin chain and a beta globin chain. Those two interacting together are going to make up quaternary structure. Some proteins only have a few polypeptide chains, making a dimer, or 3 polypeptide chains, making a trimer, or 4, a tetramer, but it could get much larger. Sometimes you've got 20 or even 50 different polypeptide chains, making one functional protein. So here's our hemoglobin protein. It's a tetramer. It's composed of 4 polypeptide chains. 2 of them are the same here in purple. Those are the alpha globin chains. And 2 of the same here in brown. Those are the beta globin chains. So that's the ribbon model of it. There's the ball and stick, and then the space-filling model. All 3 of these are showing the same protein, or the same functional protein here. It just depends on how you want to view it for which type of model you might choose. Right, so proteins are produced in the cell, but they're impacted by the surroundings. So if the environmental surroundings of the cell change, for example, the pH changes, or suddenly these proteins are dissolved in oil rather than water, their three-dimensional folding is also going to change. That can disrupt the protein's function. So that's why when you cook proteins, or like when you fry an egg, the conformation of those proteins changes. It unfolds the protein and then refolds it in a new conformation. That new conformation is no longer functional, but it makes that harder, more solid structure of the egg white that you're familiar with. Right? Depending on the environmental change, this change could be permanent, like cooking an egg, or it might be temporary if it's just a slight pH change that when it's reverted can go back to its original state. So you can also have mutations in the DNA sequence that result in a change in the protein as well. These changes are permanent to the structure. One really well understood example is what happens to the mutation in this amino acid right here. So in a normal hemoglobin protein, the glutamate amino acid, number 121, which is this guy here in red, interacts with the positively charged amino acid lysine 17 to help fold the hemoglobin protein. But you can get a mutation in this glutamate 121, changing it to a lysine so that this interaction can no longer occur. So it was a charged interaction. So an ionic bond used to form here, but if the glutamate gets mutated to a lysine, that ionic bond can no longer form. In fact, those two amino acids are repelled from each other, and then it winds up changing the overall structure of the protein. So here on the left, you have the normal structure of the protein. And on the right-hand side, you have the version with this new sickle cell version where that mutation has occurred. So this mutation in the sequence of the gene winds up changing the sequence of the protein or the primary structure. It also affects the sequence of the secondary and tertiary structure as well, changing it from this blue shaped structure more towards this red shaped structure of the beta subunit. As a result, this winds up also affecting the quaternary structure, meaning that the shape of the hemoglobin protein overall has been changed. And finally, this affects the function of the protein because the molecules would normally not associate with each other and would float freely. However, this new change in the quaternary structure of the hemoglobin protein attracts one hemoglobin protein to the next, creating this crystallized structure, which is not only less good at transporting oxygen, but also makes these fibers. So this affects, as a result, also the shape of the whole cell. Normally a red blood cell is shaped kind of like a donut that doesn't have a hole going all the way through the middle. It's like sort of a saucer or a bowl. But with the mutated version of the hemoglobin protein, the cell forms this sickle shape or this curved structure instead. These curved cells wind up not going through the bloodstream as well and as evenly and easily, so sometimes they cause blood clots because of their abnormal shape. All right, so here you can see how primary, secondary, tertiary, and quaternary structure can alter the function and can impact the function of a protein. Hemoglobin's a really good, but also very dramatic example, but this is true for any protein that gets folded incorrectly. It might not make the whole cell shape be different, but it certainly will affect the function of the protein. All right, so I hope this walkthrough was helpful. If you want to look at anything in more detail, I've posted the link for the simulation or for this tutorial in the notes so that you can find it. All right, enjoy.